Biochemistry of Cancer. The term cancer is derived from Latin cancerum, Sanskrit Greek carcinoma, carcinicum, crab. All cancers are multifactorial in origin. They include genetics, hormonal, metabolic, physical, chemical, and environmental factors. Most human cancers are spontaneous. All cancers originate from one aberrant cell which goes on to multiply and produce tumor mass. Mutagens, any substance which increases the rate of mutation, can also enhance the rate of incidence of cancer. All carcinogens are mutagens. Example: X-rays, gamma rays, UV rays, chemical tobacco of toxins, etc. Incidence of common neoplasm. In India, in developed countries, cancers in male: oral 30%, pharynx 10%, esophagus 6%, lungs 30%, colon and rectum 25%, oral 2%. Cancer in female: cervix 30%, oral 18%, breast 16%, breast 20%, colon and rectum 25%, cervix 10%. So in India, what we see in cancers in male is mainly due to oral cancer, and cancers in female is due to cervix cancer in India. And in developed countries, cancers in male is due to lung, and cancer in female is due to breast cancer. Some chemical carcinogens: polycyclic aromatic hydrocarbons, aromatic amines, nitroso compounds, natural compounds, benzopyrenes, colanthracenes, dimethylbenzanthracene, N-methyl 4 azobenzene, dimethyl nitrosamine, aflatoxins. Anti-mutagens: vitamin A and carotenoids are shown to reverse precocious conditions. Vitamin E acts as antioxidant, preventing damage made by free radicals and superoxides. Vitamin C prevents cancer in person working with aniline. Curcumin in turmeric is known to prevent cancers. Low protein, low fat diet decreases the risk of cancer in animal studies. Flavonoids are phytochemicals that possess anti-mutagenic properties. Phenolic compounds are effective in fighting mutagens found in grapes and walnuts. Green tea is effective against smoke-induced mutagens. Oncogenic viruses. Another etiological factor of carcinogenesis is the integration of viral genes into host DNA. Viruses producing tumors in animals. Virus, nucleic acid of virus, host, and type of tumor produced. Papovavirus group: SV40, papilloma, Marek retrovirus type C, Gros Rouse retrovirus type B, Bittner. SV40 DNA. Host is mouse, type of tumor produced is sarcoma. Papilloma, nucleic acid of virus is DNA, host is rabbit, and type of tumor produced is papilloma. Marek, DNA, chicken and lymphoma. Gros, RNA, mouse leukemia. Rouse, RNA, avian sarcoma. Bittner, RNA, mouse mammary tumor. Human oncogenic viruses: Epstein-Barr virus (EBV), Burkitt's lymphoma (BL), nasopharyngeal carcinoma, human papillomavirus (HPV), uterine cervical carcinoma, hepatitis B virus (HBV), hepatoma, hepatitis C virus HCV, hepatocellular carcinoma, human immunodeficiency virus HIV, Kaposid sarcoma. Oncogenes. Oncogenes are normal constituents of cells. These are genes capable of causing cancer. To distinguish viral gene and cellular gene, they are denoted VSRC and CSRC respectively. The oncogenes present in normal cells are also called as proto-oncogenes. Today, more than 100 human proto-oncogenes are known. Some cellular oncogenes. Here you can see Tyrosine kinase receptors for epidermal growth factor, transforming growth factor, DNA binding protein, platelet derived growth factor, tyrosine kinase, and GTPase. Plasma membrane, membrane, nucleus, membrane, cytoplasm. Location in human chromosome of ABL is 9, ERBB 7, ERBB A17, MYC 8, CIS 22, SRC 20, and RAS 12. Growth factors. Many oncogenes act through the production of growth factors. These may be considered as local hormones. There are more than 100 growth factors. Interleukins and interferons are growth factors released by lymphocytes and macrophages. Oncogenes and oncogenes are different. Oncogene is the chemical which produces cancer. Oncogenes are the chemicals that produce cancer. Oncogene is the gene causing cancer. Oncogene is the chemical which produces cancer, and oncogenes are the chemical that produces cancer. Oncogene is the gene causing cancer. Oncogenes are the genes causing cancers. Oncogenes are written with small letters and anti-oncogenes are written with capital letters. The gene present in normal cell is named with prefix C to show that it is in the cell, whereas the corresponding gene present in the virus is denoted with prefix V standing for viruses. So some important growth factors: epidermal growth factor EGF, chromosome number 17, molecular weight in kilodalton is 6, produced by location. Fibroblast submaxillary glands stimulates epidermal and epithelial cells, transforming growth factor TGF alpha 5.6, tumor cells placenta similar to EGF, transforming growth factor beta, inhibition of fibroblasts, platelet derived growth factor accelerates wound healing, nerve growth factor, submaxillary gland, growth of sensory neurons, insulin like growth factor, serum, sulfation into cartilage, erythropoietin, kidney stimulates erythropoiesis, granulocyte macrophage colony stimulating factor GMCSF, endothelial cells and T cells stimulates granulocytes, monocytes and megakaryocytes. Granulocytes colony stimulating factor, endothelial cells and fibroblast stimulates granulocytes. Monocyte colony stimulating factor, endothelial cells stimulates monocytes. Tumor necrosis factor alpha, monocytes necrosis of tumor cells, proliferation of leukocytes. Difference between normal and tumor cells: doubling time. It can vary widely between 10 days to 450 days with a mean of about 100 days in tumor cells. So here you can see embryonal tumors, malignant lymphomas, mesenchymal sarcomas, squamous cell carcinomas, adenocarcinomas. Contact inhibition. This property is lost in cancer cells. Anchorage dependence. Malignant character is the loss of anchorage dependence in tissue culture system. Most cancer cells carry more negative surface charge on their cell surface than their normal counterparts. This is due to higher NANA and acetylnuraminic acid, sialic acid content of cancer cell membrane. Cancer cells have a tendency to disintegrate from main mass and to disseminate to nearby or distinct organ. This is called as metastasis, most feared property of the cancer. Cancer cells have increased and persistent activity of telomerase, that enzyme that maintains the length of telomeres. Increased activity of telomerase. Tumor markers. These are also called as tumor index substances. Tumor marker or tumor index substances. They are factors released from the tumor cells which could be detected in blood and therefore indicate presence of tumor in body. They are useful for following purposes: for follow-up of cancer and to monitor the effectiveness of the therapy, to facilitate detection of cancer, for prognosis, for detection and to monitor. Tumor markers or tumor index substances are used. Precautions. Tumor markers are sometimes elevated in non-malignant condition. When a marker is used for cancer screening or diagnosis, the physician must confirm a positive test result by using imaging studies, tissue biopsies, and other procedures. Common tumor markers are oncofetal products, alpha fetoprotein, AFP, hepatoma, germ cell cancer, carcinoembryonic antigen, CEA, colorectal, gastrointestinal, and lung cancer, carbohydrate antigen, CA125, ovarian cancer of epithelial origin, tissue antigens, tissue polypeptide antigen, general cancer load, enzymes, alkaline phosphatase, ALP, bone secondaries, placental type, ALP, Regan, lung and seminoma, prostatic acid phosphatases, prostate cancer, prostate specific prostate cancer antigen PSA, neuron specific enolage, neuroendocrine tumors, hormone and their metabolites, beta HCG, choriocarcinoma, calcitonin, medullary thyroid carcinoma, big ACTH, lung oat cell cancer, vasoactive intestinal peptide, epidomas, amine precursor uptake and decarboxylation, amine precursor uptake and decarboxylation, vinyl mandelic acid VMA, pheochromocytoma and neuroblastoma, hydroxyindole acetic acid carcinoid syndrome, tumor catabolic products, hydroxyproline, bone metastasis, serum proteins, immunoglobulins, multiple myeloma, macroglobulinemia, Ben Jones proteins in urine, multiple myeloma. Ben Jones proteins, multiple myeloma. Anti-cancer drugs. Common anti-cancer drugs: methotrexate, 6-mercaptopurine, 6-thioguanine, cyclophosphamide, phosphatamide, mitomycin, antimycin D, vincristine, vinblastine, adriamycin, etoposide, camptothecin, cisplatin, imatinib, fluorouracil. Methotrexate is folic acid analog, and 6-mercaptopurine and 6-thioguanine are purine analog. Cyclophosphatamide, alkylating agent, mitomycin, antibiotic, and actinomycin D is antibiotic. Vincristine and vinblastine are alkaloids from Vincia rosa, adriamycin, anthracyclines, etoposide, podophyllorotoxin, and cisplatin and imatinib. Cisplatin platinum compound, imatinib is monoclonal antibodies, fluorouracil is pyrimidine analog. Competitive inhibitors of dihydrofolate reductase (DHFA) is required for nucleotide synthesis, and this leads to the folate deficiency or functional deficiency of DHFA. Inhibits the conversion of IMP to AMP. Inhibits the conversion of IMP to AMP. Cross-linking of basis of DNA. Inhibition of strand separation. Cross bridges are formed between DNA base pairs. Intercalates with guanine bases of DNA, prevents transcription, interferes with assembly of cytoskeleton, and inhibits stathomokine's spindle movement. Topoisomerase mediator breaks in DNA, stabilized topoisomerase 2 DNA cleavage complexes, modified function of topoisomerase 1 to DNA breaking agent, forms intrastrand DNA adducts, tyrosine kinase inhibitor, inhibits thymidylate synthase. Thank you.